Man, we have received an alert about counterfeit drugs in the market.
Attention everyone, we have an FDA critical emergency. Do you have more information about the drug?
It is an anti-cancer drug called Bevacizumab. There's a chance that few medical facilities have already purchased a counterfeit batch.
The Bevacizumab has entered the market already. It can't be that thing. We have no time to waste. Investigation group, go find the producer.
Man, what will be the instructions?
Contact the police and use all of our resources. Based on our source, this counterfeit drug has been produced by one of the university labs that have all of the technology to mass produce the counterfeit drug. It is also possible that this drug is being spread into crisis zone, which made it very deadly. We need to investigate further. Go and arrest the criminals now. We must deal with the issues as soon as possible, otherwise cancer patients will be at great risk.
Good afternoon. I have received the inquiry on the counterfeit drug case, and I'm sorry to hear such news.
Thank you for offering your help. I heard that you might provide us a solution.
Yes, hear me out. Upon your request, we have developed a technology specifically targeting Bevacizumab antibodies in the form of a rapid test.
You might be familiar with the fact that there are several classes of molecules which can be used as targeting tools, a most prominent example being antibodies. However, antibody use is limited to physiological conditions and their shorter shelf life does not align with our project's objectives. Instead, aptamers can be used as a synthetic alternative. Aptamers are short DNA or RNA oligonucleotides which can be designed to selectively bind to a molecule of choice. Their structure is more easily modified and their storage conditions are not that strict, which is why aptamers were chosen for the purposes of the project.
Yet there was another decision to be made. The most common way to obtain aptamer sequence is systematic evolution of ligands by exponential enrichment, known as SELEX. Despite being widely used, it is a timely process that has a chance to yield poor results. The urgency of counterfeit drugs should be addressed immediately, and we therefore decided to use a bioinformatic approach instead. Luckily for us, we did not have to start from zero, as there is an existing sequence already published in scientific community. That sequence has relatively poor affinity for the Bevacizumab antibody, and a decision was made to improve the affinity of an existing sequence by engineering interactions not previously involved in antibody binding.
Our methodology could then be outlined as follows. First, modification of existing aptamer. Second, obtaining tertiary structures of aptamer and antibody. Third, performing molecular dynamics simulations. We began our bioinformatics work by varying nucleotides in the existing sequence, ending up with 24 possible new aptamers. For subsequent modeling, Bevacizumab antibody initial coordinates were obtained from protein data bank. Tertiary structures of aptamer sequences were obtained using the 3D DNA web server by Yi Xiao lab. First attempts to perform molecular dynamics using GROMACS yielded suboptimal results and proved to be inefficient. We have then decided to adopt a slightly modified methodology proposed in Nature paper, resulting in an addition of an extra step, virtual screening for binding affinity.
Normalized results from 3 different models, including Autodoc Bina, Batchdoc, and HEX, identified 7 best aptamer candidates for further simulations with molecular dynamics. At the end, only aptamer 20 indicated a statistically significant results.
In order to experimentally assess the affinity of our aptamer, enzyme-linked oligonucleotide assay, or ELONA, was employed. In this assay, aptamers bind to specific target molecules, resulting in colorimetric readout, with the detection being facilitated through enzyme-linked conjugates. Basic principle of ELONA is illustrated in this diagram. During the binding phase, an aptamer candidate is incubated in a 96-well plate and allowed to attach to the surface, followed by the addition of Bevacizumab antibody. The detection is achieved by addition of horseradish peroxidase conjugated neutralpherin, and the tetramethylbenzidine solution is added last to produce a colorimetric response, measured at 450 nanometer using a microplate reader. Intensity of the signal then allows to analyze aptamer performance.
Simultaneously, we also chose to perform microscale thermophoresis, also known as MST. Recent studies in the Journal of Molecular Sciences and Journal of Materials Chemistry highlight the efficacy of MST in studying aptamer-antibody interactions in diagnostics. MST allows for the quantification of binding interactions in real time, providing valuable insights into binding kinetics and thermodynamics.
Using a combination of these techniques allowed us to assess the binding of our aptamer to Bevacizumab antibody. To begin binding affinity assessments, we have first performed Ilona and MST assay on previously existing aptamer sequence. For Ilona, we conducted a series of experiments to validate the method of chromogenic signal detection and establish a lower limit of Bevacizumab detection.
In our results, we observed that DNA aptamers demonstrate high stability against factors such as heat, organic solvents, and acids. By maintaining proper washing protocols and preserving the three-dimensional structure of the aptamers, a strong linearity of the calibration curves was achieved with correlation coefficient being larger than 0.98.
However, lowering the concentration of Bevacizumab in samples resulted in a reduction of the calibration curve slope and increased variance in absorbance between Bevacizumab samples. Despite this, the coefficient of variation values across all concentrations remained below 30%, demonstrating that some accuracy was maintained. The lowest detectable concentration was determined to be 5 nanograms per milliliter.
After MST assay performed on the existing aptamer, the cessation constant Kd of 11 nanomolar was obtained under pH of 6.3.
The results are comparable with the results of a previously published study with Kd of 12 nanomolar and the pH 7.4 obtained from Ilona assay. To note, pH of 6.3 was chosen to closely mimic the clinical conditions of Bevacizumab solutions.
Having followed the same protocol with our new aptamer, we have determined Kd to be 10.4 nanomolar and a pH of 6.3. Though the Kd of our new aptamer determined through MST is better than that of the existing aptamer, the difference is not statistically significant. Therefore, Ilona was also performed on our new aptamer for comparison. In contrast to existing aptamer, the graph for the new one shows a significant improvement in both the trend line and overall sensitivity. The normalized absorbance values increase more sharply with increasing Bevacizumab concentrations, demonstrating better discrimination between lower and higher concentrations.
Observed improvement in aptamer sequence in both sensitivity and precision indicate that the optimization applied were effective. New aptamers demonstrate superior performance in detecting Bevacizumab across the concentration range, making it a more reliable and sensitive tool for the application. This result highlights the potential of using in silico techniques such as molecular docking and dynamics to improve aptamer design for enhanced biosensor performance.
We then immobilized aptamers on the lateral flow assay strips, a user-friendly and cost-effective solution for rapid on-site testing. LFA strip design includes a control line that ensures test validity and a test line that becomes visible when the target molecule is absent.
This simple yet powerful tool is designed for easy use by healthcare providers and even patients worldwide, ensuring safety of medications. The lateral flow assay strip portability and affordability make it ideal for deployment in various settings, from hospitals to remote clinics.
All this would not be possible without expert advice. Our consultations with specialists have helped us keep our project on point and incorporate useful improvements. At first, we met with Professor Kwok from City University of Hong Kong, who gave us insights on the use of aptamers. Then, we met with Professor Ong from the National University of Singapore, who shared his experience in developing lateral flow assays. Without their help, we could not have developed the express test. Additionally, we talked with a doctor from Armenia and a medical student from Indonesia, who shared their experience in encountering counterfeit drug cases and drug shortages in those developing countries.
To bring our idea to life, we have prepared a business proposal with the help of Prof. Oleg from Department of Accountancy, who is an expert in business consulting, and Prof. Lieberman from University of Notre Dame, who specializes in design of paper analytic device to solve drug-related problems in low-resource settings. Their experience and input allowed us to describe our project in a suitable manner for startup grant application, which we plan to submit in the near future.
Moreover, we have engaged in conference to alert stakeholders and shared our innovations with IGEM worldwide. This includes Suzhou China IGEM Conference, 2nd IGEM IGA Forum, Hong Kong Macau Symposium, APEC Symposium, and more. We've also held synthetic biology workshop for future generations, sharing our knowledge from kindergarten to high school students. We collaborated with Hong Kong Children Foundation to give underprivileged children a chance to learn synthetic biology, the Cornell College to help high school students learn synthetic biology in an engaging way, and the United Christian College of Kowloon East to inspire the students about the practical use of the synthetic biology.
Aside from that, we try to connect synthetic biology with art to attract and engage the public. We have worked with the KCIS Hsuagang IGEM team in Taiwan to share synthetic biology innovations in the form of postcards for local Taiwanese students. We have also made a biome picture book from the McGill University IGEM team to illustrate DNA-RNA interacting with proteins. Moreover, our own team is making a children's synthetic biology storybook with fun illustrations and a synthetic biology-themed board game. We hope through this artistic pursuit approach, we could attract more people from various backgrounds and ages.
To further help the community, we have made several education materials to be shared and used. We are making an education biosensor video with the Washington University team and to inspire the society about the use and importance of biosensors. And meanwhile, to help other scientists conducting research, we've collaborated with Queen's University IGEM team to make research ethics handbooks. And not only handbooks, we have also given a talk about research ethics directly to the community with Nazarbayev University's IGEM team. And lastly, we are developing a synthetic biology activity handbook to guide teachers and students to do simple synthetic biology activities in class.
Dear SDA, don't move.
We have a warrant to search your laboratory. Hope for your cooperation. All of you are under arrest for the production and sales of counterfeit drugs.
What are you talking about? We just work in our lab.
We found enough evidence. Send sample to Bevatech now.
You have the right to remain silent. Anything you say can and will be used against you in a court of law.
Today, we are pleased to announce that thanks to the incredible capabilities of the Bevatech device, we have been able to identify and dismantle a significant counterfeit drug operation. Its accurate and affordable technology enabled us to rapidly and accurately identify counterfeit drugs. Thanks to Bevatech, our commitment to safeguarding public health has never been stronger.